simulations to functional design, and before I will go directly to the results of our lab, let me say a few words. Why do we actually need to design a drug delivery system? What is drug delivery systems? And why do we actually care about it? Well, the development of drug nanocarriers represents the critical focus of pharmaceutical research for more than 20 or 25 years. Nevertheless, this interest is still very huge because it is driven by the need to overcome the limitations which is associated with conventional, let's say, drug formulations and enhance the therapeutic efficiency of the drugs. And when we have drug in free form, it may be characterized by poor solubility, short circulation time, non-specific distribution in the organism, and so on. And to overcome these limitations, drugs are used to be encapsulated within the specific carrier, typically of nanometer size, and this results in numerous advantages, which include enhanced drug solubility, improved bioavailability, protection against degradation, etc. And in the context of precision medicine or personalized medicine, such drug delivery platforms are very critical for integrating the diagnostic and therapeutic functions within one single system. And this is called theranostics. I hope that a lot of you heard this term, and this field, theranostic field, these directions currently developing very, very rapidly. And up to date, a lot of different drug delivery systems have been developed, but among them, the system which is based on protein molecules represents a very promising class because of their biocompatibility, biodegradability, and the ability to interact specifically with some biological targets. And to fully understand the potential of protein-based nanocarriers, of course, we will start with computational methods, the results of which surely should be confirmed by experimental techniques. But as far as you understand, the first step at the designing of protein-based drug nanocarriers will be computational simulations. A lot of different computational techniques such as molecular docking, molecular dynamic simulations, coarse-grained modeling enable detailed insights into drug binding mechanisms, into protein conformational changes when we encapsulate some drug molecule within it, and the stability of the overall nanocarrier platform. Several simulation techniques may, let's say, have leading positions. And among them are molecular dynamic simulation, of course, which models the movement of atoms and molecules in the system over a certain period of time. Monte Carlo simulation is very promising in the development and design and the drug delivery systems, which uses random sampling to simulate the behavior of drug molecules in a system. Molecular docking, of course, the approach which allows to predict the localization of the drug molecule within the protein and to identify the binding sites. Finite element analysis, very informative technique, and it uses no, it not uses, it actually models the behavior of drug delivery systems under different conditions using a set of different mathematical equations for better understanding of the mechanical properties. Of course, such modern approaches such as machine learning and artificial intelligence-based approaches are currently very rapidly developed in the field of in the field of the design of protein-based drug nanocarriers. In my next slides, I will briefly overview what have been done by first. I will overview what have been done by different researchers using various simulation approaches to design drug delivery systems, and then we will discuss what we are doing in this aspect. As I said, molecular dynamic simulations, let's say, represents the leading position in drug carrier design. It is very informative tool for investigating the fundamental mechanisms which governs protein drug interactions and structural stability of the resulting complexes. Molecular dynamic simulation represents a computational technique that studies the behavior of atoms and molecules over the time. And the typical molecular simulation algorithm is illustrated on the slide, and actually it doesn't differ from from the molecular dynamics of any other particular biological system. The technique is based on classical mechanics, as you know, which describes the behavior of particles in terms of their position, momentum, and forces which act on these particles. Fundamental theory of molecular dynamic simulation is based on a motion-based equation, which are derived from Newton's equations, and pretty good information we can extract from this type of simulation. Different laboratories and a lot of researchers have extensively employed molecular dynamics simulation to elucidate the binding mechanisms between different therapeutic agents, different drugs, molecules, and protein carriers. And these results revealed critical insight into the intermolecular forces that drive drug encapsulation and release. For example, computational studies of soy protein as a drug delivery system demonstrated that electrostatic interactions dominate the binding of polar drugs, while pp staking interactions are crucial for aromatic drug molecules. These simulations have provided very detailed understanding and very detailed mechanisms of how protein denaturation processes can enhance drug loading capacity by exposing additional binding site. In addition, molecular dynamic simulation have been very informative in understanding the design of ferritin-based drug delivery systems. So these studies demonstrated that such drugs like anti-malarial drugs can be readily incorporated into ferritin without disrupting the native conformation of ferritin, while when the researchers incorporated antiviral drugs, the conformation of ferritin was broken, and this was like selective binding of the drug molecules to the protein. Next, comprehensive molecular dynamics studies of transferrin have elucidated the complex mechanisms which underlie the receptor-mediated drug transport across different biological barriers. And the researchers applied combination of molecular dynamics with homology modeling, and they predicted the complete three-dimensional structure of the resulting protein-based nanocarrier. Molecular dynamics simulations were also employed to investigate the behavior of different polymer complex, different peptide polypeptide actually complexes, which are different in their side chains, and it was concluded that for lactic acid. Here it is. Lactic acid is the most promising variant among the analogs in this polypeptide row. Next type of simulation which can be used successfully in the design of protein-based nanocarriers is Monte Carlo simulation. This is a computational technique that uses random sampling to simulate complex systems and different processes which occur in these systems. The technique is widely used, of course, in various fields, as you know, not only in prediction and design of protein-based nanocarriers. And the basic idea behind the Monte Carlo simulation is to use random sampling to generate many potential outcomes or different scenarios for a given particular system. Monte Carlo simulation can statistically estimate the behavior of the system by simulating many potential outcomes, including probability distribution and expected values. And of course, this technique is very informative in predicting the structure of protein drug complex and its stability. For example, Monte Carlo simulations was used to model the release of doxorubicin from protein nanoparticles of different size and composition. The authors represented nanoparticles as discrete voxels and simulate polymer degradation and drug diffusion, and the results that they obtained very accurately predict multi-phase drug release without this result actually allowed the researchers to avoid very hard measurable physical parameters that should be that should utilize very complicated experimental techniques, and pretty fine results were obtained. Coarse-grained simulations in their turn also were informative for predicting drug protein systems. These methods reduce system complexity by grouping the atoms into the larger bits. And this enables simulation of bigger assemblies and longer time scales relevant for drug delivery nanocarriers, like, for example, lipid-based nanocarriers, which is liposomes or protein aggregates. For example, Martini-based coarse-grained simulations have been employed to study the drug encapsulation behavior into albumin nanoparticles of different size, and they revealed that environmental factors like ionic strengths or pH affect the size of the resultant nanoparticle and the stability of this particle, which is, of course, very critical for effective drug delivery. This very brief, very discrete examples show that information that can be obtained using different simulation approaches is very diverse and very informative. But now I would like to outline the results of our research in this area and to concentrate on particular specific examples that we have obtained based on our results. And the first example is development of nanocarriers for such compounds as polyphenols. You can see the structures of the examined polyphenols in this slide. Polyphenols represent the natural compounds which are found in plants and possess a lot of different activities, including antioxidant. This is the main, actually, their role in the organism, and they contribute to the prevention of various diseases. And clinical application of polyphenols is very limited due to their very low bioavailability and rapid metabolism in the body. And one of the solutions to this problem is, of course, the development of polyphenol delivery nanosystem, particularly those that is based on the protein molecules, which, as I said at the beginning of my lecture, may overcome these limitations. We applied molecular docking and molecular dynamics approaches to evaluate the principal possibility, let's say, of creating such protein-based nanocarriers. In the protein components, we used albumin, insulin, and cytochrome C. Analysis of here is the structure of the complexes of protein polyphenol complexes, which we obtained from molecular docking simulation. Uh, Analysis of the best score values obtained for the complexes of polyphenols with different proteins revealed that all polyphenols possess the highest and comparable affinities for albumin and hemoglobin, while the lowest affinities was observed for insulin. If we check the table, you may see that the lowest affinity for all polyphenols that we studied actually was observed for insulin. After molecular docking, the stability of all complexes was evaluated using the molecular dynamics over 1000 nanosecond time scale, and it appeared that not all complexes were stable, despite the fact that molecular docking revealed that all complexes are formed and a lot of different interacting amino acid residues were identified in protein molecules which are in contact with the polyphenols. Molecular dynamics showed that not all complexes appeared to be stable, and this, of course, compromised um, the further development of such complexes as drug delivery carriers. Furthermore, the analysis of protein conformation after the drug encapsulation showed that some polyphenols have an impact on protein structure, which may affect the effectiveness of the resulted drug carrier system. Because, of course, if we encapsulate polyphenol into the protein and the protein breaks down after some time of interaction, the idea, the initial idea of using this protein as a drug carrier that will protect the drug and that will enhance its bioavailability, this idea will be also broken down. So based on this integrative results from molecular docking and molecular dynamics simulation, we identified the complexes which were stable over the whole time and scale range, and which showed the best docking scores, and it found that albumin represent the most promising carriers for polyphenols. Next, we evaluate the feasibility of creating protein-based nanocarriers for different combinations of polyphenols, specifically for binary and ternary combinations, using once again albumin and insulin as carrier molecules. To achieve this goal, we conducted a three-step docking, multi-step docking. First, the protein was docked with gallic acid. Then additional docking was with another polyphenols to form either binary or ternary combinations. And according to the data which are presented here in the table, albumin once again showed the best choice, and we concluded that it may serve as an effective carrier for both binary and ternary polyphenol combinations, because once again the complexes with albumin were stable over the whole time scale range, over the whole molecular dynamics simulations, and best token scores was also observed for hemoglobin. And here is the interactions between different combinations of polyphenols and proteins when we identified the complexes that show the best results and that can be, let's say, recommended for further development. We analyzed what amino acid residues of the albumin are involved in the complexation with the polyphenols. and it is clearly seen that hydrogen bonds represent the predominant forces, the predominant bonds that stabilize the protein polyphenol structure. However, another interactions such as aromatic or hydrophobic are also present, but for all complexes, this hydrogen bond, which is highlighted here as green, it represents the main force for stabilization of polyphenol protein complexes. And an albumin, okay, I will skip this, I will say about this. Um, unlike albumin, when we the next example of our research is in the development of protein-based different systems for the transport of different drugs. Included anti-COVID pharmaceuticals. And these systems were based on lysozyme. And here we tried to design the so-called theranostic protein-based nanocarrier. So the systems that will include both therapeutic and diagnostic modalities. And to achieve this theranostic potential of the system, we combine the therapeutic agent, actually the anti-COVID drug and the visualizing agent, which was represented by fluorescent drug within the one protein system. And to evaluate the principal possibility of such combination and to check the stability of such complexes, we conducted a series of molecular dynamic simulations, which was based on top scored docked protein complexes, and the molecular dynamics results have been analyzed in terms of different parameters such as backbone, root mean square deviation, duration radius, etc., etc. And for some systems, we observed that the detachment of either the drug or the fluorescent dye molecule occurs, and this means that such complexes cannot be recommended for further development, but some systems appear to be very stable and very effective. Among all combinations studied, the most promising was found to be the combination of a drug which is called ritonavir. Here it is designated as letter R, and here it is highlighted in blue, and cyanine dye, which is called AK75, which is here as green, and these two structures, these two compounds simultaneously encapsulated within the lysozyme, and these complexes were found to be the most stable among all systems studied. Here is the parameters that were derived from molecular dynamic simulations, and that confirms our conclusion. Next example deals with the development of multimodal systems that integrate therapeutic imaging and targeting components. One of the most promising combinations for multimodal drug delivery systems is the integration of radionuclides and fluorophores. Radionuclides are the molecules that are used for visualization of different oncological tissues, different oncological structures and formations within the body using specific techniques such as positron emission tomography. But radionuclide per se may represent the therapeutic agent, and radionuclide per se has a very huge radioactive effect on the organism, and it is very important to control the distribution of this radionuclide within the organism and the accumulation of it within the tissue, the oncological neoplastic tissue. And this goal, the idea was to incorporate once again the radionuclides and fluorophores into one multi-facet toolkit, and to generate the multimodal drug delivery systems that can achieve both therapeutic and imaging functions. And we actually succeeded to conduct such design, such preliminary evaluations. What we have done, we use antineoplastic drug doxorubicin. We use two visualizing agents, radionuclide and fluorescent dye, and incorporate all of this stuff into the protein carrier that was represented by albumin to achieve to generate the theranostic multimodal platform. Doxorubicin represents the therapeutic agent and serves for treatment the oncological processes, and this multimodal visualizing agents are necessary to complement the results of imaging of each other. Incorporation into albumin results in the increased bioavailability and increased therapeutic index of the resulting system. For this, for construction of such system, we used a lot of actually 10 radionuclides which are based on technetium-99M, and as fluorescent dyes we used four. Among them was the cyanin green, methylene blue, and two dyes which do not have specific names. It's like only the production name, but this is squalane dye, and this is cyanin dye. And at the first step, we compared the binding affinities of different radiopharmaceutical for albumin, and here is shown the best score complexes of different technetium structures with the albumin. Polypeptide chain of albumin actually contains a lot of amino acid residues. To be more strict, it contains 585 amino acid residues with three homologous domains. Domain number one, this includes residues from the first up to 195. Domain number two, amino acid residues up to 383, and domain number three, the rest of the structure. And each of these domain, here is actually domain number one, number two, number three, and each of these domains consist of two subdomains. This subdomain one, subdomain two, and it is generally recognized that the majority of drugs bind to albumin through the two sites, which are known as Sudlow, site 1 and site 2. Why actually albumin is so important in the designing of protein-based nanocarriers? Because albumin represents the main plasma protein, and it has long circulation time in plasma. And of course, if we encapsulate the drugs or visualizing agent into the albumin, we may expect that the circulation time of the resulting system also will be prolonged, and we will have a lot of time of operation of such system in the organism. And when we docked different radiopharmaceuticals to the albumin, we found that the binding site for almost all radiopharmaceuticals represent domain number one, which is not common for typical drugs. At the next steps of the investigation, we employed the multiple ligand docking approach to explore the ternary protein ligand systems, which include actually protein per se, radionuclides, and antineoplastic drug doxorubicin. And this studies the structures actually of such complexes are presented here, and the main conclusion actually that was driven from these studies is that there is no competition between doxorubicin and radionuclides for the binding sites on the proteins, and this is actually very important because this may give the hope that the stability of the system will not be compromised by the combining by the competition binding and by the specific interactions between doxorubicin and radionuclides inside the protein molecule. So this specific interaction may disrupt the structure of albumin and may break down the whole structure of the carrier, and then the idea of such complex will actually not be so good as it may seem at the first. But since no competition was found, we concluded that the structure of such complex may be stable, and we continue investigating the potential of this complex. And then the next step, we try to obtain the systems with dual imaging modality. Uh, Once again, the structures of the resulting complexes, which contained everything that we could have, I mean, radionuclide, fluorescent dye, and antineoplastic drug doxorubicin. Here is the best score complexes, the best docked structures of the complexes, and the comparative analysis of the docking results obtained for such quaternary systems revealed that the best affinity was for the dye, which is called indocyanin green, and this allows optimizing the structure of dual modality imaging systems. Of course, these obtained results are very preliminary and require further corroboration, and we have actually the problem when we try to conduct the molecular dynamic simulation of such systems, because the technetium being metal, a heavy metal, it doesn't appear in force fields for molecular dynamics, and this is actually a big problem which we are trying to overcome, but unfortunately we have no progress up to now in this aspect, but we are continuing to work in this direction. Actually, I think that I will stop now because we are out of schedule. And if you have any question connected with our specific examples of our research, please, I will be glad to uh, answer them.
Thank you very much, Valeria, for your talk with very interesting background and very interesting research. While I open the session for the discussion, I have some curiosity. You presented many different study with a nice combination between docking and molecular dynamics, and it was very interesting to see that the molecule that might look promising with docking, then in the light of molecular dynamic simulation, they are not so much. And you, with these studies with the polyphenols, you show that albumin is the most promising protein. And I was wondering, what are the next step of your study? Are you planning, when possible, to check this? How?
Thank you for your question. The next step of our study is we are planning to actually to conduct the experimental validation of the stability of such structures, and to actually to make it experimentally in vitro, and to check where the stability is such as was predicted by computer simulations.
And do you have in mind some specific experiment like NMR or I don't know, circular decoys?
Of course. We will. We are planning actually to use the whole set of techniques that is available for us to check to check the stability of the complex, the structure of the complex, and to check once again whether the predictions made by computer simulation are in harmony with experimental results. But the main idea of this computer simulation was to recommend the complexes, to identify those complexes that may be promising, and then to check it experimentally, because we have no possibility to create experimentally all the complexes that I have discussed, all proteins with all polyphenols, because it is very time consuming and it is very expensive, will be very expensive procedure. And once we have identified the complexes based on albumin, now we will be working experimentally in this direction to prove that our computer predictions and computer estimates are corroborated by experimental observations.
Thank you very much. Another curiosity, what program did you use for the docking?
Actually, we use the basic program is Autodoc Vina, but we basically usually we checked the docking by different servers like CBDoc, SimDoc, but the basic docking program is Vina, Autodoc Vina.
Okay, I have other curiosity, but I let now the other people ask their question. So please ask the question. I don't see the room now for the visualization mode I have. I don't know if there are questions, Sergi, from the room.
In the room, actually, we do not have questions at the moment. So probably I saw Sonia raised hand. Yeah, we have Maria Pilar raised hand.
Okay, so please, I don't know who answered. I will raise the hand first. So let's start with Sonia, and then in the order that Sergey said. So please, Sonia.
Sonia, we can't hear you.
Okay, I'm here. Thank you, Valeriya, for this very nice and inspiring talk. So you mentioned that you have some problem with force field. So I'm just curious, which force field did you use for this the last slide when you speak with this multimodal therapeutic system?
Actually, we are conducting our molecular dynamics in GROMACS, and we have encountered the problem that no technetium in we tried every force field we knew, but no technetium is written for molecular dynamics, because as far as I understand, the chemistry of our organisms is not based on such heavy metals like technetium, and that's why it is very difficult to compute it. The idea was to change technetium with rhenium like as a chemical analog, but we are currently in the process of this research.
Okay, so modeling the systems with metals is really not easy.
Yeah.
Since you have to define also the geometries around the metal and this melodic.
Yeah. I'm sorry, I don't know why I cannot switch on my camera, because
just a moment. We have to ask to enable camera. Yes,
Yeah, I
probably.
see that you are asking me, but the option to switch on the camera
Ask to
is
start video. After start video, yeah. Please push.
yeah, but the option the button to start my video is not active. It's inactive.
This is some technical issues that we have
Okay,
today.
let's.
We try to fix
Okay.
it probably tomorrow, but unfortunately today we have such problem. I don't know why it's happened.
It's not a problem. I'm just sorry for not being on the screen, only my portrait.
This is our fault. Sorry. So the question was about Sonia asked.
The question was about, yes, technetium and molecular dynamics, and Sonia said that it is a tough task to conduct actually the molecular dynamics with the metals, and actually, yes, we
Yeah,
understood.
this is
Yeah, this is one of the challenges for the force field development when metal is present.
well. Actually, I have
Yeah.
easier question. Probably, sorry if I interrupt, but this
Okay,
is
thank you,
just to
Valeria.
this question. Why technetium? So this is so rare metal. Actually, this is exotic metal as for me.
This metal is one of the most used for as a as radioisotope for radiopharmaceutical development, because it is. This is technetium is a radio isotope, and it is currently used in nuclear medicine for visualizing the different tissues with oncological states, and
I see. This is probably I missed this. Okay, thank you.
yeah.